So good day everyone, we are the Itchyworms and we are going to have a quick review session for the lab exam focusing on nematodes. The goal of this review is to help you refresh your memory on the major characteristics used to identify nematodes, especially the one commonly found or the specimens we have in the laboratory. So beginning with station 1, we can observe one image and with the following questions of scientific name, life stage, what sample can the specimen be found in, and if the specimen strictly requires two separate organisms as a host. So to answer these questions all combined, we have Trichinella spiralis coming from the coiled appearance of the larval stage, which is actually observable in this picture. And this larva is found in muscle specimens or muscle tissue, given that round circular pink structure around it, which is the muscle cell, which the organism uses for protection and feeding. And lastly, this specimen does not require two separate organisms as a host, as it only needs one. Moving on to station 2, we have two images wherein we have to identify key structures along with key characteristics of each image. To answer these questions, here we have two adult stages of Trichinella spiralis, with its feeding structures being the stichocytes, with males having a distinguishing feature of copulatory clasps on the posterior end, with males also appearing much shorter and much thicker than females. On to station 3, we have an entirely different species, along with images representing it. Here we identify key stages along with its descriptions about them. So to answer this, image X is actually a rhabditiform larva, while image O is a filariform larva. The infective stage is in fact the filariform larva, with the non-infective stage being the rhabditiform larva. Both specimens can be found in fecal samples. In station 4, we will finally discover the scientific name of the specimen, along with its key structures and characteristics. So this species here is actually Strongyloides stercoralis, with the image showing an adult worm with its gender being female. This is because males are not considered parasitic. This parasite causes strongyloidiasis, a disease which can be greatly aggravated by people with immunocompromised patients. Moving on to station 5, we have an organism with two completely different life stages wherein we have to identify key structures and characteristics. The organism we have here is Trichuris trichiura. Its egg in image X has polar plugs which are found in opposite ends of the egg. Its common name is the whipworm due to its whip-like anterior end. Image O is considered a male due to its coiled appearance in its posterior end, which actually contains its reproductive organs. Moving on to station 6, we have a new species with three different images wherein we have to find key structures along with key characteristics. The organism here is Enterobius vermicularis. Its defining feature in its egg stage is its slightly flattened appearance in one side. It has a pin-like tail, which can only be found in males, and image W is actually the longer female. Its main and only host organism is humans, or better known as Homo sapiens. Moving on to station 7, we have two very distinct images of actually the same life stage. Now the organism we have here is Ascaris lumbricoides in both its egg stage. So image X is actually the unfertilized egg while image O is the fertilized egg. This is because image O is round and oval with a well-defined embryo and a thick shell, while X is more elongated and narrow and contains a mass of disorganized, refractile granules rather than a clear embryo. So station 8 seems to consist of an image of a cross-section wherein we have to identify a series of pointed structures. So station 8 is actually a cross-section of a female adult Ascaris lumbricoides. Structure A is specifically its cuticle, structure B is its intestine, structure C is its uterus which contains its eggs, and structure D is its ovary, further proving that it is a female. So station 9 consists of an entirely different species, wherein we have to identify its life stage and its key characteristics. So this organism is Onchocerca volvulus. Image X is its microfilaria stage, while image O is its adult stage viewed as a cross-section within subcutaneous nodules. Its intermediate host is the Simulium fly, and the infection actually causes blindness, specifically the disease river blindness.
Now let us move on to question 10. For station 10, the specimen appears to be Loa loa, a filarial nematode transmitted by deer flies of the genus Chrysops. The diagnostic stage we see here is the microfilaria found in peripheral blood smears. The red arrow indicates the sheath surrounding the microfilaria, while the blue arrow points to the row of nuclei extending along the body and reaching the tip of the tail. This characteristic helps distinguish it from other filarial parasites. The specimen causes loiasis. The infection is characterized by calabar swellings, which are transient localized swellings in the subcutaneous tissues, and the migration of adult worms across the conjunctiva of the eye, giving the parasite its common name, the African eyeworm. Now we move on to question 11. The specimen that we see here seems to be Dipetalomena sp, a filarial nematode that infects humans as a definitive host. Some species previously classified as Dipetalonema are now included under Manzonella. Here we see that number 1 is the oral opening, while number 2 is the esophagus, a muscular tube in the interior region, and number 3 is the vulvar opening. And so overall these structures help us distinguish that this is a female adult worm. Next, we see here that number 1 seems to be the left spicule, while number 2 is the right spicule, and letter C is the cloacal opening. So since these structures are part of the male reproductive system, this worm is classified as male. The male is actually much smaller than the female. The microfilariae that we see here is unsheathed, which helps us differentiate this specimen from other filarial worms such as loa loa. Infection with this parasite causes manzanillosis, a usually mild filarial disease that can affect subcutaneous tissue or body cavities depending on the species. Next, we move on to question number 12. Here, panel A seems to show us Manzanella perstans, which is transmitted by biting midges of the genus Sulisoides. Panel B shows Manzanella ozarti, which is transmitted by black flies of the genus Simulium. Panel C shows Manzanella streptocerca, which is also transmitted by biting midges of the genus Susisoides. The nuclei extend to the tip of the tail in specimens A and C, which correspond to Manzanella perstans and Manzanella streptocerca. Manzanella perstans has a blunt-shaped tail, Manzanella streptocerca has a hook-like shaped tail, and Manzanella ozarti has a tail bent in a small hook-like shape. These differences in tail morphology help distinguish the species under the microscope. These parasites also cause manzanillosis. Next, we move on to station 13. For this station, the specimen appears to be Capillaria sp or C. philippinensis, a nematode that can affect the intestine or liver depending on the species. Humans are the definitive host, where the worms may produce eggs that are passed in stool. The diagnostic stage we see in A here is the egg. The eggs are peanut-shaped with bipolar plugs at both ends, which is a characteristic feature that distinguishes Capillaria from other nematode eggs. In image B, the female is indicated by O and is slightly larger than the male. She has an esophagus, which is number 2, that is muscular tube about half the length of her body, a vulva, number 3, which is the opening for egg release, and a uterus, number 4, which contains developing eggs. The male is indicated by X and is slightly smaller, with a small caudal alley and a spicule with sheath, as indicated in number 1, which is a thin, spineless reproductive structure used during mating. Next, we move on to number 14. Specimen are Trichostrongylus sp, an intestinal nematode that primarily affects herbivorous animals such as sheep and cattle, although humans may become accidental hosts. The diagnostic stage observed in A is the egg, which is thin-shelled and elongated, and yes, it is tapered at one end, a feature that helps in microscopic identification. These eggs resemble hookworm eggs, although Trichostrongylus eggs are typically slightly larger and more elongated. The blue arrow indicates the rudimentary and unarmed buccal cavity, which is a characteristic feature in Trichostrongylus adults. The answer here is false, because Trichostrongylus does not possess cervical papillae, helping it be differentiated from other nematodes. Next, we move on to question number 15. The specimen that we see here is Angiostrongylus cantonensis, a parasitic nematode commonly known as the rat lungworm, with rats serving as the primary definitive host. Based from panel A, we can see here that the male is labeled as number 2 since it is typically smaller than the female and has a more slender body. Meanwhile, the female is labeled as number 1 since it is generally larger and thicker compared to the male. So, let us now label all the parts in panel A and B. Number 1 indicates the buccal capsule, while number 2 is the esophagus, number 3 points to the excretory pore, and number 4 indicates the genital primordium. Number 5 represents the anus, and lastly, number 6 indicates the pointed tail. For the last question, yes, the egg is unembryonated, meaning that the embryo has not yet fully developed inside the egg.
So moving on, here the family is being asked, and based on the morphological features, they must be part of family Filariidae. Next, what specimen is different among the rest? So I hope most of you got it correct. It's specimen X since it is Wuchereria bancrofti. Specimens Y and Z are actually just the same species, which is Brugia malayi. So if you base it on their distinguishing features, we can see that in Wuchereria, their body nuclei are equally sized, clear, and countable, while that of the Brugia are unequally sized, uncountable, and coalescing. So these specimens are all actually of the microfilaria stage, which is the diagnostic stage in humans. Since specimen X is Wuchereria, its vector is a mosquito. Another point if you put the genus, which is Culex, although it can also be Aedes, Anopheles, Mansonia, and Camelatidia. For letter D, true, since one of the distinguishing features of Brugia, although not seen here, are the terminal nuclei in tandem with each other, which is, on the other hand, absent in Wuchereria. Lastly, for letter E, all of them, both Wuchereria and Brugia, causes filariasis. Next station, fill in the table and compare the morphology of Wuchereria bancrofti and Brugia malayi. Size-wise, Wuchereria is larger than Brugia. In terms of cephalic space, as we have seen in the previous slide, that of Wuchereria is shorter while Brugia's is longer. For the nuclei, as we have seen a while ago as well, Wuchereria is uniformly sized, clear, and countable, while Brugia's is unequally sized, coalescing, and uncountable. Wuchereria species have no terminal nuclei, while Brugia have two terminal nuclei that are in tandem with each other. Both have the same L3 filariform infective stage, while both also have the same transmission and diagnostic stage, which is the microfilaria stage. Here we can just see a clearer labeling of the morphological features of the two species. Seen are the difference in body nuclei, the presence of the terminal nuclei in Brugia malayi, and the difference in length of the cephalic space. S stands for sheep. Here are the life cycles of the two species, which are just actually the same. The definitive host is infected with the filariform larva of the mosquito vector. The larva then matures in the host and produces sheeted microfilariae, which are then ingested by the mosquito in its blood meal. The microfilaria then matures in its thoracic muscles until it becomes again the L3 filariform larva, which then migrates to the mosquito's proboscis for the next infection. Take note that these worms do not have free-laid eggs since they already develop into sheeted microfilariae in utero.
For the next 4 questions, I'll be discussing about the reproductive types of nematodes. I'll give you a quick second to answer the following questions. Okay, so let's answer the following questions. For A, the correct answer is H because hermaphroditic worms typically possess both the female and male reproductive system in one individual and are self-fertilizing. For B, the correct answer is P for parthenogenetic because this typically describes a virgin birth where the egg does not require a sperm to be fertilized. For C, the correct answer is O for oviparous because the hatching of eggs typically takes place in an external environment. For D, the correct answer is OV for ovoviviparous because before giving birth, the eggs are already hatched inside the uterus. And then for E, the correct answer is true. So for this number, we will be identifying the type of reproductive system of specific specimens that were already discussed in the earlier numbers. So I'll give you a second again to try to answer the following questions. Okay, let's go over the questions. For letter A, it is famous for being a parasitic parthenogenetic female in the human gut. The correct answer here is L, which is Strongyloides stercoralis. For B, which of these is ovoviviparous and releases larvae into the mucosa? The correct answer here is H, which is Stipiralis. For C, true or false, Enterobius vermicularis, seen here as K, is an oviparous nematode, which is true. For D, I is an oviparous nematode that produces lemon-shaped eggs with polar plugs. What is the name of the species? The correct answer here is Trichurius trichuria. And then for E, which of these produces unsheathed microfilariae? The correct answer is J, which is Onchocerca.
So now for this station, the specimen shown is a hookworm egg. It is oval in shape with a thin smooth shell. Inside the egg, we can observe segmented blastomeres or a morula stage, usually around 4 to 8 cells. An important diagnostic point is that the eggs of the two major human hookworms look identical under routine microscopy. So in stool examination, they are usually reported simply as hookworm eggs. The two species responsible for the most human infections are Ancylostoma duodenale and Necator americanus. These eggs are typically passed in feces in an unembryonated stage, which serves as the diagnostic stage in stool examination. Moving to the next station, let's review the life cycle stages. If arranged from youngest to oldest, the correct sequence is egg, rhabditiform larva, filariform larva to adult worm. Among these stages, the filariform larva is the infective stage. Humans become infected when these larvae penetrates the skin, commonly when walking barefoot on contaminated soil. For the larvae to develop successfully outside the host, they require warm, moist soil conditions. This direct life cycle is characteristic of soil-transmitted helminths. Here we differentiate the two main hookworm species based on their buccal capsule or mouth parts. Figure A represents Ancylostoma duodenale. This species has 2 pairs of ventral teeth in its buccal capsule. These teeth allow the worm to attach to the intestinal mucosa and feed on blood, which can lead to iron deficiency anemia in heavy infections. While for figure B represents the Necator americanus. The statement that both species have teeth is false. Necator americanus does not have a teeth. Instead, it has a ventral cutting plates in the buccal capsule used for attachment. Now for this station, we observe adult Ancylostoma duodenale showing sexual dimorphism. Specimen A is the female while specimen B is the male. The female is generally larger than the male. At the posterior end of the male, we can see the copulatory bursa, which is used during mating. In contrast, the female possesses a vulva, which is absent in the male. The anterior end also shows the buccal capsule with teeth, which is used for attachment and feeding. Similar from the last station, this species is again Necator americanus. Note that the sexual dimorphism presented here, figure A is the female while figure B is the male. The green circles highlight the mouth or buccal capsule, while the copulatory bursa at the posterior end of the male is used for reproduction. These features help us distinguish sex and species during identification. Since we have already reviewed their mouth parts and body shapes, focus on the remaining diagnostic features. Note that Ancylostoma duodenale possesses a caudal spine and a circular copulatory bursa, while Necator americanus lacks the spine and has an oval bursa with a different vulva position. Okay, now for this station, aside from the human hookworms, there are also animal hookworms that may infect humans. One example is Ancylostoma brasiliense, which normally infects dogs and cats. When humans are infected, the larvae usually cannot complete their life cycle but instead migrate within the skin, causing cutaneous larva migrants. Other zoonotic hookworms include Ancylostoma caninum and Ancylostoma selanicum, which may occasionally infect humans. So now, based on the figures in the table, which species is the most distinct? Necator americanus is the most distinct species, while the four other species of Ancylostoma possesses various arrangement of teeth. Necator americanus is unique because its mouth capsule features ventral cutting plates. Again, true or false, all species shown in the table have humans as their definitive host. Again, it is important to remember that not all Ancylostoma species use humans as their definitive host. Other zoonotic hookworms normally infect animals, but may also occasionally infect humans.